So Alexander's going to talk about Soma and SomaLogic and SomaScan in the first 10 minutes of the talk. We're going to get a lightning talk from Barry Z. And then Lindsay and I are going to focus on the main part of this session, which is going to be on alternative proteomic technologies, sort of where they stand currently, and from a mass spectrometry perspective, how can we best interact with them and relate to them and use those as useful technologies to ourselves. In a sense, the theme of this is that there's more than enough work to go around for all of us to actually be able to get useful data, very important data. So with that, I'd like to let Alexander start talking about Soma.
Hello, everybody. I'm Alex Scalia, and I'm with the SomaLogic arm of Standard BioTools. So I'm going to talk today about the SomaScan platform. So to get started, for those of you who might be new to SomaLogic, we're a leader in measuring proteins and identifying biomarkers. So the flagship product of the SomaLogic platform is the SomaScan assay. The assay has been validated not only by ourselves, but globally by other groups and other partners of ours. And collaborators have put out over 600 publications. The leading proteomics assay is available as both a service via SomaLogic or is available to set up in your own labs. So we've been around for over 2 decades. We got started in 2000 and really have just ramped up the content on our menu ever since then. And we really see the utility of the technology through the publications and the citations that have come out of those. They really indicate, as we open up our menu and expand into new protein territories, the researchers are finding ways to be able to use that technology. So most recently, we expanded our menu from 7,000 to 11,000 assay measurements. And what we did with this during our menu expansion is we really tried to open up to proteins that are really representative of the proteome itself. So we didn't want to just measure any old protein that's around. We wanted to give some sort of an indication representative of all the different biological pathways and all different diseases that use these pathways. So the SomaScan assay itself is built on the core technology of a SOMAmer reagent, and this stands for a slow off-rate modified aptamer. So how we build these reagents are from single-stranded DNA with amino acid-like side chains. These amino acid-like side chains allow for the DNA strand to behave as protein-like as possible while staying small and being reproducibly manufactured. So what these modifications end up doing is they add structural motifs that allow for the shape of our SOMAmer reagents to maintain and really stay consistent. And they also add to the binding interfaces between our SOMAmer reagent and the protein to which it's binding. The DNA synthesis process is done within our own laboratories outside of biological systems, and that is really reflected in the CV that we see. It's very low, around 5%. So the affinity that we end up seeing because of the way that we develop our SOMAmer reagents, we used lessons learned from antibodies, and because of that, we're able to achieve similar affinity to what the antibodies have. And we start off to create these reagents with a pool of about 10 to the 15th unique sequences, and we run it through a selects process to really refine those sequences into allowing the winners to really just show themselves to us. And this exquisite scalability or specificity really gives us the opportunity for a very scalable platform. We've actually noticed the CV improve as we've opened our menu up and increased the size. So one of the challenges of the proteomic assays that we run into is the really huge dynamic range in the endogenous levels of proteins in the human body. So we run our assay through a 3-dilution series where we're able to measure both the high and low and middle abundance proteins all at the same time without any enrichment or manipulation of the samples. So what this allows us to do is we know we can measure the low abundance proteins without risking affecting them by looking behind those high abundance proteins. So the SomaScan assay itself is relatively simple once you break it down. So we start off by immobilizing our SomaMer reagents, the entire 7 or 11,000 menu, onto magnetic beads. We call this catch 0. Then we take the sample, and in the 3 dilution bins I mentioned earlier, we allow them to bind to the SomaMer reagents. So through this, we end up with some specific binding and some non-specific binding. We then biotinylate the proteins, we zap them with UV light and release the photocleavable linker, allowing these SomaMer protein complexes to float into solution. We then add our polyanionic competitor, which is a very promiscuous mid-affinity protein, or SomaMer, and we allow this to outcompete the non-specific binding interactions. So what we end up with in catch 2 is capturing our proteins via the biotin from step C, and we have proteins with SomaMers in the specific binding interactions captured, and proteins with no SomaMer left over from the non-specific binding interactions. We wash away anything unbound, elute off the SomaMers, and then measure those on a Agilent microarray. So we only end up measuring the SomaMer reagents from the specific binding interactions, which gives us a much better idea of the actual representative proteins in the original sample. And broken down to its most simple form, what we did was we turned a very difficult problem of measuring proteins, and we kind of converted that into measuring DNA, which is something that we know how to do. So I mentioned our microarrays. Those are from Agilent. We partnered with them. So the way that these work is that they print out the complementary DNA sequence from each SomaMer. Oh, I guess it went away on me. And those are put on probes in specific locations on the slides. So then, based on the location of the lighting up of the probe, we can see the location tells us which SomaMer reagent and therefore which protein we're looking at, and then the intensity of the light tells us how much. So then within the services that we offer, we break these out into 3 categories. So the first is the SomaScan assay, which we run at our headquarters in Boulder. So scientists, researchers, anybody who wants to work with us, they can send us samples and we run these samples in-house for them. The second is our SomaSignal tests. So these are clinical risk assessments that have taken a look at a subset of clinical samples, and we've developed tests that can, based on the protein profiles of these samples, really be used as diagnostic tests. The coolest one, in my opinion, is the pre-analytical variation, which takes a look at proteins that are more and less sensitive to things like time to centrifuge and freeze-thaw cycles. And then we have our SomaLogic authorized sites. So these are opportunities to set up SomaScan in each of your own labs and run the samples yourselves with the protocols, buffers, and equipment that we give. We run a variety of different samples. We develop our SomaMer reagents to human proteins, but depending on the sequence homology seen between species and between sample types, as long as the protein's intact, we can measure it to some degree. With different species, the sequence homology between them will be an indicator, but we released menus on both mice, pretty much any of the animals seen here. So through all of this, we have an authorized site with high-plex proteomics, and I'm running out of time, so I'm going to breeze through the rest. And the SomaScan assay really leverages the high specificity of our reagents to be able to multiplex and do these assays within a low sample volume. We run into about a 5% reproducibility through the CV and incredibly high throughput with a great dynamic range. And I don't have time for questions up here, so we have a booth in there later. Please come by and chat with us a little bit. Now I'll pass off to Barry Z.
Oh, here, actually, let me give option for one question.
Oh, can you hear me okay?
Yes.
Okay. Can you explain how plex and throughput, that interaction, how they scale with each other? Is there a dependency?
Yeah, so as we increase the plex of the assay itself remains within the same steps. So we create a master mix of these reagents. So we have all 7 or 11,000 reagents put into one assay, and we run that entire assay on the samples themselves. So when we go from the 4,000 reagent increase, the assay itself doesn't change at all. So what we end up seeing is that our throughput is based on specific binding interactions, so there should be no interference from any non-specific interactions. As the menu increases, we're still only working with specific binding interactions.
Great.
All right.
All right. Thank you.
Thank you.
So I'm just going to shout while Barry, you want to set up your slide. So I just want to introduce Barry Z from Searle Signaling Technologies. He's going to give a brief lightning talk, and then we'll talk more about some other technologies as well.
Let's see. Oh, just seeing how this works. Is that this thing right here?
Yeah, that's good.
Just make sure. Okay. Well, great to see everyone here. My name is Barry from CST, and really happy to tell you about our work on post-translational modification analyses. So over the past few years, we have developed a number of antibody magnetic bead conjugates that are targeted against various PTM classes independent of the surrounding amino acid context. And one of the great things about antibodies is their antigen specificity, and we can see that in this table here, sort of comparing our first product, ubiquitin lysine, to our most recent product, pan-methyl-lysine. There's very little cross-reactivity or enrichment for other PTM types. Now, because of our antibodies, we designed them to recognize PTMs independent of the surrounding amino acids. This basically means that these antibodies recognize peptides independent of the species. And they all can follow a pretty similar workflow, which is we simply bind the peptides, wash the peptides, and then elute them off the beads. Now, the promise of antibodies is that they provide quantitative and scalable recovery of the enriched PTM peptides across a reasonable range of input material. And here we've demonstrated from about 1 milligram down to about 100th of a milligram. And so we get about a few hundred PTM sites, and each PTM is going to be a little different in how low it can go. But the other feature, the great thing about magnetic beads, is that we've demonstrated compatibility of that workflow manually, so on a bench in a magnetic rack, but also across various automation platforms. Here's an example with a Kingfisher, and on the graph illustrating that we get comparable performance, whether you perform it by hand or on the instrument. So we're really excited about the opportunities that these antibody bead conjugates would do for PTM analyses. So.
So while I get started, can you guys raise your hands if you use a mass spectrometer? Okay. Can you raise your hands if you use a proteomic technology that is not mass spectrometry? Can you raise your hands if you only use a proteomics technology that's not using a mass spectrometer? Cool. Okay, good. So we're predominantly mass spectrometrists in this room, and of the people that are speaking today, predominantly mass spectrometrists. And so when Pratik invited us to kind of give a session on alternative proteomics technologies that weren't mass spectrometry, I was kind of like, well, what do we do here? And so what we chose to do is, I think, give something that's a little bit different than just sort of getting a parade of different technologies up here. And we designed the program to be a little bit different, and that is to focus from a mass spectrometry perspective, what are these alternative technologies, kind of like illustrated in a laundry list, one after another, what their advantages are, what their disadvantages are, what new technologies are coming down the pipeline. And then Lindsay is going to talk in the second section about how do mass spectrometry perform well or poorly at specific types of tasks? And where are circumstances where using alternative technologies might be a good complement to mass spectrometry? And then how do we kind of merge forces rather than fight a war against other technologies? And then lastly, how can we show the relevancy of each of the individual components of proteomic technologies going forward? Okay? So I'm going to talk specifically about alternative proteomic technologies that currently exist and what kind of new technologies are going to be coming down the pipeline. And so the first thing that I'm going to start is talk about Western blots, something that I hate terribly and was the reason why I got into mass spectrometry in the first place. So a Western blot would be a circumstance where you're using an antibody as a readout for a protein abundance. And so there's two ways to think about this. So you can run run a gel, for example, or a blot, and we can stain specifically for an antibody and see it light up at a certain abundance in each of the different lanes, in each of the different, for example, cell lines in this particular example. Now, one of the reasons for having this be a two-pass circumstance, this is a beautiful antibody right here, is that most antibodies look kind of like this one over here on the left. And so in this particular case, this antibody has it has specific binding ability for the particular protein that you're interested in, but it also has a lot of non-specific binding interactions as well. And so there's 2 points of evidence here, right, that are used to make the assessment. There is both the antibody binding and then the molecular weight of the protein as it's running through the gel. And so oftentimes we forget about these different aspects, the fact that it's a two-part assay, and that both of those parts are important for making the assessment, because usually when we're reporting these in the literature, they end up looking like this, right? And they look exactly the same no matter what you're looking at. And this is particularly important when we think about circumstances where we're working with a single antibody binding event. So one example of that is IHC, right? Immunohistochemistry is a circumstance where you're washing a particular protein across a cell or a tissue slice, and you're trying to see what parts of that cell light up. And so you can get really specific binding, or what looks like really specific binding to areas that just are not the protein that you're actually interested in with these types of measurements, because you are working off of a single binding event, right? A single selection event, okay? So how are different ways of getting around this sort of single binding event issue? One of the most common ways to try to think of this is trying to think about it from the standpoint of, these types of antibodies will bind to a specific event. When we're thinking about alternate approaches for making measurements, a really common one is an ELISA, specifically a sandwich ELISA. In a sandwich ELISA, you immobilize a particular protein, you wash over your proteins of interest, and then you apply a secondary protein that's binding to a different location. And that secondary protein will have a reporter. You'll read out the reporter as a way to say how much of this protein is actually present in this arrangement. So again, we're getting around needing to use a molecular weight, running a gel, because gels are annoying. And we're using a secondary antibody as an alternative, right? So there's, again, 2 selection points for doing all of the measurements to say that we believe this to some reasonable degree. Okay, so now let's go through some of the technologies that exist in the field currently that we're, as mass spectrometrists, potentially competing against or working with in those circumstances. So one of those would be, for example, Olink. So Olink basically is trying to take this idea of ELISAs and multiplexing it through using RNA sequencing. And so the idea here is taking 2 antibodies and tethering to them DNA strands, right? So when you get ligation of these DNA strands, when they're in proximity with each other, so if we have those DNA strands coming together, we can get a readout of this specific sequence, right? And that readout of that specific sequence gives us an idea of how much of that protein is actually present. This is pretty similar to Alex's approach with Soma on just trying to use DNA sequencing or RNA sequencing as your readout of a proteomic measurement, right? And so Olink is going to be doing this with a two-binding event, essentially parallelized ELISAs. And the idea here is that each one of these sequences can be parallelized because you can label each one of them with a reporter region, right? So each binding event has a reporter because you sequence that section, and each section of double-stranded DNA can also have ligated to it a barcode for that particular sample. So you can do this actually in quite high throughput, right, with multiple sets of samples simultaneously, multiple sets of proteins simultaneously. And so this is an alternate way of trying to analyze a proteome, again, using two binding events to measure that specific protein. So I don't need to talk that much about Soma because Alex has talked about this already, but the idea basically is you have your proteins around, you have your SomaMers. Your SomaMers are these modified DNA sequences, and you essentially wait for a binding event, you pull down using streptavidin or some other binding reagent, you'll pull down the complex of the DNA with the protein, you'll release it, you'll do a competitive assay where you're adding in a competitive reagent, and then you'll recapture the protein, and then what you read out at the end is the sequence that was used in the SomaMer itself. And so one point here is that we're using a single binding reaction here, right? As opposed to a double binding reaction with Olink or with ELISAs. So this is in some ways more analogous to that IHC example that we talked at the very beginning. Now, is this better than a single antibody? I would say it's probably equivalent to a well-vetted antibody, and maybe not as, like, maybe a poorly vetted antibody is worse than this. But is it equivalent to seeing paired antibodies for the same protein? It's not equivalent to that, right, in terms of its specificity. And we'll get to specificity again at the very end. But the other aspect of this is that in both of these cases, for both Soma and for Olink, we're essentially doing a single molecule binding event is each of our measurements, right? We're getting a DNA sequence barcode associated with each one of our binding events, and we tally up those barcodes, right? And that's our measure of intensity in these cases. And so each one of those is a single event that we're measuring with our sequencer at that particular time point. And we'll come back to that idea as well. So for other types of technologies, there are several other ones. So those are the kind of like the big two that have been in the news a lot lately. But we should talk about some of the other technologies as well. Protein microarrays have been around for a very long time. And there are many different varieties of protein microarrays that I think are viable at this point. One approach for doing protein microarrays is just to stamp down antibodies onto a glass slide, wash over proteins, and see binding, just as if you were doing a microarray from Affymetrix, 20 years ago. Again, same kind of constraints that you have in that circumstance with washing and all that sort of stuff and making sure your quality is good. Typically, these assays can work around 1,000 proteins, as opposed to SomaMers, where there's, I think, 11,000 which were reported, and Olink has somewhere around 5,000 to 6,000 proteins that are being used. So there are other ways of thinking about microarrays as well, and there are several different groups that are doing this to a degree. One way is to actually try to plot down proteins instead, or binding reagents, antigens instead, and then actually trying to see what binds to those antigens, right? And so one great example of this is with antibody arrays. This was incredibly important for us to understand how the immune system was fighting COVID, for example. We would actually take the COVID spike protein, we would digest it up or only print out, like get 11 mers or 13 mers, which would get stamped down on a slide. We would wash people's blood over those, their antibodies over those, and then see kind of whether they had reactivity, and then what areas of the protein did they have reactivity. And that's what led to the vaccine creation. So these are really powerful techniques for being able to make measurements of the proteome, or at least parts of the proteome. In this particular case, the sort of the autoantibodies that are generated in an individual. So protein microarrays are a powerful technique for doing some specific tasks like that. And I also want to bring out the idea of RiboSeq, right? So RiboSeq is a technology that's been around for a long time as well. When I first saw RiboSeq, I thought, oh, our community is dead. We're in such trouble. And turns out, actually has not come to pass. And I believe that we could take lessons from that going forward, right? We've already seen this type of disruptive technology before. So with RiboSeq, basically, you have all of your RNA that has been created, and the ribosome will bind to different sections of the RNA. And if you were to sort of see what was actually being read by those ribosomes, that would be representative of what was actually being created at that specific time by the cell, right? So it's not a measurement of the RNA, it's not a measurement of the protein, it's a measurement of what is actually being translated at that specific time point. And so the way this type of assay works is you've got a single-stranded RNA sequence, and you've got all of your ribosomes that are attached to it. And so the first thing you'll do is you'll chop up all of that RNA, and then you'll wash away all the proteins. Sheltered in the region around the ribosomes is around 27 nucleic acids. And so from that 27 nucleic acids, that's around 9 amino acids. That's the equivalent of a peptide that we would be measuring in proteomic measurements. It has the same sort of selectivity as a short peptide with proteomics. And so you can read those sequences as a proxy for what is actually being translated at that specific time. This gets over kind of like the major hurdle with proteomic analysis using RNA-based work, which is that RNA doesn't actually represent what's going on in the cells at any given time point, right? It represents maybe what that cell is trying to do, but it doesn't represent sort of what's changing in that cell. So if you look at the half-lives, right, so the turnover of specific proteins on the y-axis here, and the RNA turnover on the y-axis, there's basically a random scatter shot, right? They have no correlation to each other at all. So using kind of like what is being transcribed isn't a proxy for what is being translated. This is a proxy for what is being translated, but only a proxy for what is being translated, which is why mass spectrometry has existed since this. Okay, so I want to talk about next what are new technologies that are coming down the pipeline, and then we'll talk about kind of vetting these technologies from different aspects, right? So what are new technologies that are coming down the pipeline that we should be aware of as proteomicists? One of them is nanopore. Oxford Nanopore has changed how RNA sequencing works in many labs because of its price point as well as its throughput. And so we expect that Oxford Nanopore or other nanopore-based methods will come about soon, and so there are several labs that are trying to propel this type of technology. So there's kind of two different strategies for trying to deal with this so far. The first one is, let me see if I can get a pointer here. You guys can see that? Okay. So the first one is trying to to take your protein of interest, pass it through this much larger nanopore than what we use for RNA-seq. And the first thing that it's going to do is it's going to encounter an unfoldase as it's entering into the nanopore chamber. That's going to unfold the protein. And it's going to pass through the proteasome, through this nanopore here. We're going to pass the sequence of it through this lipid bilayer, and in that bilayer, we will measure the current that is changing as a result of that particular nucleic acid, or that particular amino acid passing through the pore, right? And so basically what's going on here is a simple like V equals IR equation, where as you're passing a new amino acid through, you're changing the resistance of the membrane, and so you pass a current, so a specific voltage across that membrane, and you measure current as your readout. And so essentially what we're seeing is this current trace over time as amino acids are being funneled through the pore itself, the constriction of the pore. So this is one solution for this type of work. This is actually a really hard problem because unlike nucleic acids, which have very simple constrained space and functional groups, it's much more complicated to try to pass through amino acids through something like this and getting a meaningful result, because amino acids above and before change the physical chemical properties of the amino acid that's currently being passed through. So this is a strategy that I'm sure is long-term going to work because of AI, currently doesn't work at this stage. Another way of trying to treat this problem is if you were to take a section of this giant pore and put a proteasome in it. One way that you could pass through a particular protein, you could digest that particular protein, like trypsin, for example, pass through peptides, and now you're working with trying to measure peptides instead of an entire protein, where the context of the amino acids in the peptide are a little bit more constrained. Now, there's a couple of different cool ideas centered around this approach. One of them is that, well, you can start stacking this nanopore to be gigantic, right? It doesn't have to be just a pore itself and just an unfoldase itself. You can start thinking about different ways of actually using the pore as machinery to start making measurements. So one of the ways, for example, with amino acids that's been proposed is to use this helicase strategy of tethering. Basically, you tether to the end of the protein some DNA, and then you feed the DNA through rather than the amino acids. That way you get a constant up and down motion of the protein that you're interested in through the pore. And you can actually do this not just one way, but you can do it backwards and forwards, and get multiple reads of the exact same protein. So there are lots of different interesting strategies for trying to deal with that. Another strategy that's been proposed is just to take your protein sequence, cut it up into amino acids, and basically do Edmond degradation in a pore. Right? Not Edmond, amino acid analysis in a pore. And so you're not getting sort of the sequence of the protein, but you're getting the amino acid composition. So any of you guys that have done amino acid analysis know that it actually works pretty well to identify particular proteins. And so here I've got a plot based on peptide length and the number of unique peptides in the human proteome here. And if you're thinking about sort of knowing the perfect sequence, if you knew exactly what that sequence was, then this would be your specificity of the uniqueness of that peptide in the proteome, right? So if you've got 5 or 6 amino acids, you don't really know with specificity what that particular peptide is, but once you hit around 7 amino acids, you know with as much specificity as you are going to know how good what that particular peptide is. Now, if you think about just doing amino acid analysis, if you had perfect quantitation, that would be that pink line. You know, it's not as good as knowing the exact sequence information, but even if you just know the amino acids that went into that peptide, you can actually do really well. And so if you don't know anything about the quantitation of those amino acids, then that would be this blue line. But if you know just even a 2-fold difference between amino acids, you actually can get really close to that perfect analysis here. So that would be this limited frequency information. That just knowing 2-fold difference, I know that I've got 1 amino acid of this type or more than 1 amino acid of this type. That's enough to identify a 20 mer or a 10 mer in this type of experiment. Okay, so that would be one technology that I think is coming down in the next 5 years. Another technology that I think we will see maybe sooner is this technology from Nautilus. So Nautilus is trying to use, again, antibodies, but rather than trying to use specific antibodies, they're trying to use the really bad antibodies, the ones that we would normally discard, right, that have very little specificity. So these bind to very small regions with not very good specificity frequently, but they all have kind of like a fluorescent tag on them. And so the idea here is that as you pass over a specific antibody around a protein that's been tethered to a specific spot in location, you can see sort of a pattern light up. And remember, these antibodies are terrible, right? So they'll light up everywhere. But they don't light up literally everything, right? So you want a bad antibody, but not that bad, right? That it binds to literally everything. So you get a pattern down here. And so the idea is once you take that pattern, after enough of these bad antibodies pass through, you can use the confluence of information of those to pass into, again, a machine learning algorithm that is going to spit out some information about the actual abundance of those individual proteins. So that is, again, another strategy for trying to deal with this. Just like how we've been talking about, both nanopores and this Nautilus technology, again, single molecule detectors. Single molecule detection is sort of the wave of the future here, or at least one way to think about it. Quantum Sci is another approach that uses Edmond degradation, essentially. So the idea is this was developed by the people that were doing sequencers back in the day, and the idea here is they're just trying to take sequencing technology and apply it to proteins. So they tether a protein down onto a slide, and then they do serialized Edmond degradation, where they say, okay, let's get a binder, again, not a great binder, but one that's specific to an amino acid, and then they say, okay, do we see fluorescence here? Yes or no? Chop off the next amino acid, pass over another reagent, see if there's sequence, and then chop it off, go over and over and over again. So that's another type of technology developed in part by the McCarty lab, but Quantum Sci has been trying to commercialize some of these approaches. The issue is binders in these particular cases. Right now, we don't actually have very good binders. The binders that were in the science paper were these 3 binders here. So they could recognize arginine. They could recognize one of 3 amino acids, L, I, and V, so isoleucine, leucine, or valine, or the aromatics, F, Y, and W, right? So those give you some information about what actual proteins are present, but not a ton of information. Okay, so single molecule detectors. So that's kind of like my laundry list of what I think is coming down the pipeline. So single molecule detection is, I think, really one of the important things that we need to talk about here with these types of technologies. Really, we want to divide up all of these technologies into whether they're single molecule detectors or multi-molecule detectors. So single molecule detecting is like reading a DNA sequence, a fluorescent pulse, sort of like that Edmond degradation, molecular reactions, right? So again, sort of like amino acid analysis, Edmond degradation. Those would be single molecule detection approaches. So Olink, Soma, RiboSeq, nanopores, the Nautilus approach, the Quantum Sci approach, these are all single molecule detecting approaches. Right? A multi-molecule detection approach is where you pile up a whole bunch of molecules of one particular type and you make a measurement of those. So those would be things like the intensity out of a mass spec, or the intensity out of an HPLC, or the fluorescence intensity of a particular set of molecules. And so these would be things like IHC or Westerns or microarrays or mass spectrometry as we typically think of this. So that separation of 2 different types of technologies, I think, is our biggest clarifying agent in trying to understand what each of these technologies is good at. And so one of the ways to think of this is with RNA, single molecule detection has been really powerful. It's been incredibly powerful for being able to measure the amount of a pileup of a specific read. And the reason why is because the dynamic range of a genome isn't that large, right? If you think about all of the RNA that is produced by a specific cell, typically you see sequences that range from 1 read out to around 1,000 reads, where the median of those reads is around 17, and it's very tight in that specific region. If you compare that to a single cell's worth of nucleic acids or proteins, the range is much, much larger, right? So we tend to see in a single cell, we'll see as great of a range as e to the 8th, right? Where the median protein amount, the number of copies of that particular protein is around 50,000 proteins. Now this is really important from the standpoint of single cell, for example, where if you're making a measurement of just 17 on average molecules, your dynamic range is kind of crummy. But if your dynamic range for single cell analysis with proteins is around 50,000, then you have a much better chance of getting a real analytical measurement using a single cell approach. But let's try to tackle that up even to a global approach, where we're thinking about kind of like the copies per cell that we're actually making measurements of. In those particular cases, every copy of this protein, and remember there's e to the 8th up here, we have to read through at least one of the, like we have to read through e to the 8th of those before we start seeing stuff down here. Does that make sense? Right? Every time you read one of these, you will have to have plowed through all of this stuff to have been able to see that low abundant thing. Now, we can use antibody binding reactions as a way to do enrichment for those particular types of signals. But remember, at any given time point, in any, like an affinity-based reaction has only get you a couple orders of magnitude of improvement in dynamic range, right? So typically what we're seeing here are circumstances where you can make measurements down here using single molecule measurements. But when you start thinking about this from a background perspective, right, most of the time we're likely to be seeing background in these types of alternative technologies. Now, in proteomics, we have a very well storied history of vetting specific detections for confidence, right? How good do we believe this specific protein detection, not just how good do we believe the confidence in the quantitation, right? In RNA world, right, typically they just measure the confidence of the quantitation, not even caring about the quality of the detection itself. So in these particular proteomic technologies, we have to remember that there is no vetting, at least at this standpoint, of how good a quality of any individual binding reaction actually is, right? And so there's no idea of a detection false discovery rate as well as a quantification false discovery rate. And so when we're trying to make these measurements down at very low values, we can get consistently measured results of background, right, in a given sample. But that doesn't necessarily indicate that it's the actual protein that we're interested in trying to measure, right? So you can still get good CVs, but it might not be what you actually think you're measuring because you might getting specific binding to background, just like with IHC that we were seeing before. Okay, this becomes really important when we start thinking about big dynamic range fluids like blood, right? So, so if you have a company, for example, that says we can identify, 10,000 proteins in blood, we all know there's not 10,000 proteins in blood, right? So you kind of have to take this with a grain of salt in that you can make a measurement of 10,000 proteins, but your quality of that measurement definitely has a different level of sophistication than what we're talking about with mass spectrometry, where we have kind of thresholds at detections as well as thresholds at quantification. Okay, so with that, I'd like to kind of conclude here. There's a wide range of these alternative technologies that are interested in challenging mass spectrometry for relevancy. Antibodies remain, and affinity reagents remain a critical part to all of these types of technologies, right? It's funny that we're going back to this idea of trying to run Westerns in IHC. I really am sad about this, but I understand that this is the direction that we're going. False discovery rates are important for us to assess. And we've spent a lot of time thinking about target decoy, for example, with respect to mass spectrometry. We have to start thinking about detection, discovery assessments with other technologies as well, in the same kind of rigor that we've done so far with mass spectrometry. So some types of experiments, I think, are better suited to using affinity-based reagents. And so we need to say, there's some problems that maybe as mass spectrometrists we actually should just step away from, let other people kind of tackle those while we dig into other types of problems. And there are other ways that we can kind of make complementary analyses. And I think that the last thing that I want to hammer home is that this idea of counting single molecules works really well when you've got a very small dynamic range. It does not necessarily scale well to proteomics, style measurements. Okay. So with that, I'd like to conclude my section. I'd like to open up for Lindsay to actually talk about, okay, well now we've talked about all these technologies, what does it mean in the context of a mass spectrometry lab, in the context of a proteomics lab? How can we kind of interact with each other better so that we can collaborate better and cooperate better? So with that, I'll take Lindsay. Great. and then we'll have a discussion at the end about kind of like this, what's going on here. Great. Yes. Are there any questions while we're swapping over? Yeah.
Yes, and we decommissioned our machine a number of years ago now because we were crippled by antibodies. And within that whole society, no one wanted to say whether a vendor's antibody was good or bad because they didn't want the retribution from that. So how do we go forwards with these new antibody-based technologies, trusting that the antibodies are actually going to be able to measure what they say they do?
Well, because we don't ever get access to the antibodies that the companies are making, or the molecules that the companies are making in the case of Soma, we don't have the ability to assess them one by one on our own, independently from the company. We do have to rely on a certain degree of trust about the quality of those reagents. Now, the companies, I would argue, are better at producing antibodies than sort of like the Abcams of the world that are just sort of selling kits for ELISAs or Westerns without any knowledge of what's going on in the background. At least these companies have taken some serious consideration about how to vet the quality of their affinity-based reagents. But we have to take that on face value at this point, right? We have to trust that they have done it well.
Yeah, that's the point, right? Like, that's exactly my point, because, you know, every antibody that's out there, how is it automatically now a position where we can, you know, I'm very interested in Soma, but I guess I'm being the devil's advocate, because how is it that there's now 10,000 available antibodies when the RPPA field is still struggling, and that field's been around for a long time? Maybe Lindsay will address this, but my next question is throughput. For the mass spectrometrists, and I don't do mass spec, but we are desperate for 384 well plate format, high throughput proteomics that can measure cytokines, for example.
Yeah,
so
I hope Lindsay is going to talk about that. That is getting a lot better, yeah. And throughput has gotten a lot better with mass spectrometry, as well as these other techniques are designed to deal with 96 well plates or 384 well plates too. Okay.
Along with that, we talk about binding to a particular protein. Classical case is BNP in plasma and congestive heart failure. So we've been measuring that for a very long time and correlating it with stage and grade. And it turns out that almost not, it's immunoreactive. There's lots and lots of immunoreactive BNP, but there's no biologically active BNP in those people. And so the proteoforms, which is invisible to us, right? How do we start trying to validate or identify what proteoforms are being detected in these multiplex assays? And I would say that there's a role for core labs to do these experiments. A consortium would be useful. The government should be funding this, because they're funding all the research that says, hey, we got this number from this assay, but we don't actually know what it's binding, and we need to make a library of what it's binding.
Yeah, basically you're opening up perfectly for Lindsay. So I think we'll leave it at that. But absolutely, different splice forms are really critical that you can't deal very well with antibody-based methods.
Yeah. Thanks, Brian. And I'm glad we're already kind of starting the discussion, because the whole point of the slides I've put together are to try to maybe put out a couple hot takes and start getting people talking. So it sounds like we're already there. That's great. Brian, in this last session, kind of focused on the technical aspects and the scientific aspects. I want to maybe speak a little bit to, as business people, anybody who's running a core or a CRO, you have a business aspect to what you're doing, right? You need customers, or else it's hard to justify why you exist. You need to manage a budget. So I want to talk a little bit about that aspect of what we do. As my conflict of interest, I'm a CTO at a startup company that I've co-founded. We run our 2-instrument mass spectrometry closet as a core facility, kind of within our internal programs, balancing that to fund our internal programs with these external programs, and figuring out how do we draw customers that'll help pay for these instruments that are incredibly expensive. So I had just kind of mentioned this, but this is not meant to be an exhaustive report of how we can run cores, how we can do business development. It's meant to inspire some conversation, like Brian was saying. And I think we all appreciate here, everyone had put their hands up at some point regarding the general term of proteomics. Some people were mass spec, some people were not, some people were both, but everybody had this interest in this work that they're doing in proteomics. And how you're doing proteomics might be specific to the core, where you're geographically located, or your expertise that you have amongst your staff. But anything from basic biology all the way through highly regulated clinical diagnostics, through food safety, and more, proteomics is being used a lot of different ways. And Brian just gave us a really great overview of all of the different technologies that you can use to measure proteins, but I would add there's even more, right? Proteomics is not just measuring of a protein, but we can consider how protein chemistry and protein measurements include other potential core facilities like a cryo-EM or biochemical assays. So let's just address the elephant in the room. Why should we still have mass spectrometry cores? Why should we still have mass spec facilities, right? We heard great talk about SomaLogic. Brian just told you there's all these other options out there which maybe have their pros and cons. Why should a mass spec facility still exist? Brian had mentioned I want to talk very briefly about where we're going to be replaced, where I think we're losing ground in that customer segment and why. And then we're going to talk about where we're, I don't think, going to be replaced. And this, again, is not an exhaustive report. This is my perspective. It's meant to fire you up and get you to the mics and start talking. And then finally, I want to talk about how we can work together as a community, this ABRF community, to build these justifications for a mutual existence of all of these technologies. It doesn't have to be a competition, it can be a collaboration. And the long story short is the proteomics total market cap is huge. If anything, we should all be working together to go replace the genomics people. I know, right? That's why I said it a little bit softer. Okay, so where are we going to be replaced? Biofluids. Biofluids. We're going to be replaced in biofluids. Throughput is the short version of that, right? Biofluids with mass spectrometry. We've got these great 96-well plate workups. People like Jenny Van Eyck over at Cedars-Sinai is running a fantastic biomarker discovery and validation core facility using mass spectrometry as a readout. It just requires, to do the throughput that a lot of biofluid studies are looking to do, it's thousands. It's not just a 386-well plate, it's multiple of them over longitudes. And mass spec just has not delivered in the throughput and the scale that biofluids researchers are looking to do. High structure proteins, where it's very easy to generate antibodies, where there's already existing or validated antibodies. These globular high structured proteins that have been very well used in a lot of other aspects that require recombinant proteins like protein microarrays or SPR, surface plasma resonance. Those types of proteins, there's a lot of other technologies because all of these other technologies work really well. And I had mentioned this kind of with the biofluids, but I think for incredibly large-scale sample sets where you have thousands of samples that need to be processed and analyzed and then reported with actionable insights, I think those are just a string of things that mass spec really fails to deliver. So these three areas, biofluids, it's every technology, and Brian talked about a lot of them, and there's a lot more. Alamar Biosciences here, there's all of the next-gen protein sequencers like Arizion. There's so many options out there, and those are just the ones we can see, right? That doesn't even include the more like stealthy startup companies that I'm sure are being developed out in Boston, in the Bay Area, and San Diego. Biofluids are the best beachhead for them to be working on and solving because it is such a high value market with a very high market cap. So biofluids, there are millions, billions of dollars being poured into other technologies other than mass spectrometry, and even our mass spec vendors recognize this, right? Thermobot Olink. Like, they know, they see what's coming, and they've already made their bets to diversify that mass spec is going to be replaced in these capacities by Olink. High structure proteins, these readily available antibodies, it's going to mean it's very easy to commercialize other technologies. And then all of these not mass spec technologies have made it incredibly turnkey for people to do large patient cohorts, and almost all of them actually have really nice analysis and reporting that we could only dream of in mass spectrometry. If I never had to spend another day putting together a report for a client on a project where I am digging through data through multiple softwares and trying to piece together what is the actionable insight I can give them, that would be the best day so far of my career. I think mass spec is not going to replace in some other areas, things like whole proteome measurements. All of these other technologies in some way or form are kind of a targeted panel. They're having to do a lot of work to add each additional protein. So a lot of them, like I had said, are focusing in biofluids, so proteins that are not present in biofluids. I think those types of studies are going to be pretty safe. Highly unstructured proteins, membrane-bound proteins, my personal favorite protein family of transcription factors. These high unstructured proteins make it very difficult for them to be worked with a technology that requires some form of structure, which almost everything does except for a mass spectrometer is pretty structure agnostic. We actually destroy all the structure before we put it into our instrument. And then finally, as we were just saying, the non-canonical proteoforms. So things like a splice variant, things like PTMs, things like truncated proteins. I think all of these are places where the versatility of mass spectrometry can be very powerful, that other technologies would have to do a lot of development to build up those kinds of panels. Our difficulty, our blessing and our curse is that there is so much informatic things you could do to either program your mass spectrometer or prep your samples a different way or report the data, analyze it a different way. That is great for these kind of ill-defined problems here, but obviously becomes a bit of a downfall when you just need to do one simple thing and do it a lot of times. So mass spec, we just heard, is sensitive for thousands to tens of thousands of proteins all at once. It's not one of those single molecule technologies Brian was talking about. So we can do a lot of things in a single sample without doing a ton of assay development work. Because LC-MS is structure agnostic and doesn't require antibodies, we can go after some of the harder proteins, which kind of makes light of how difficult doing membrane proteomics is, but it is much more accessible with a mass spectrometer than other technologies. And then because a mass spectrometer is just measuring the mass, it's inherently going to be tolerant of a lot of these non-canonical isoforms. Now, if that isoform is somehow the exact same mass or fragments the exact same way, we're going to have difficulties, but much less difficulties than some other technologies. So some places we're going to be replaced. Some places I think we're going to be kind of ingrained as the leading choice for some proteomics approaches. And I think one thing I would like to put out there as a conversation starter is that some of these more disruptive or competitive technologies are actually fantastic for us as mass spectrometrists because they are raising awareness of proteomics that didn't exist before, or that people are still thinking about proteomics as it was 20 years ago, where it was like Nature Method of the Year and these targeted protein assays, and then it kind of didn't really work out. Then there was that whole controversy with the dinosaur proteins. So people are maybe kind of still thinking about proteomics that way, and having these new technologies is bringing them back to considering, oh, maybe proteomics is a good choice for what I'm doing. This opens up new markets that haven't traditionally come to proteomics and new prospective customers that we can start thinking about targeting as a community. So a rising tide is going to lift all of these boats. Instead of thinking about trying to split the wedge of proteomics pie across all of us, I would encourage us to think about just the pie is getting bigger. So your wedge can stay the same or grow bigger if we're building this proteomics as a whole community, collaboratively instead of competitively. So that was kind of elephant in the room. Why should mass spec facilities still exist? And two other conversation starters that I'd encourage us to think about is kind of the business development aspect of what we do, not just the science and the technology like we were talking with Brian, but also the business side of our roles. What are the value propositions? Who are the stakeholders? And in these slides, which we'll figure out how to disseminate at some point. There's some questions that if you've had some business development courses, either in your formal education or just as you've been growing in your role, you'll notice come from the business model canvas, which is a free PDF. You can just Google that a lot of new companies and new directors have to think about. The first, understanding these stakeholders. You know who your customers are. You know who the customers are, probably of the other cores or facilities that are available at your institutes. But you can start to think about this in the context of what if there was, if there isn't already, what if there was a SomaLogic or an Olink or a Nautilus or an Alamar being offered in competition to your services? What are the channels through which you're advertising your core? Can you think about other channels to be advertising your core? If you're only internal so far, how can you start to think externally or working with your administration to get some of these external customers? Reviewing kind of not only how you're reaching them now, but how you might reach them in the future, and what are some cost-efficient ways of doing that? And then that's kind of the people side of this. I'll wait for the picture, yeah. And then the other side of it is the value proposition. So this is the question we can be asking ourselves, kind of this exercise of what is mass spec doing really well? What is mass spec really failing at? And part of this can go back to the stakeholders, going back to collaborations or projects or customers that you've had in the past and asking them, like, what worked well for you in this project, in this statement of work? If you were to do another SOW, what would you like to see differently? So we were talking about this, Barry and I, about how like a product team and a services team really have to work closely together. If all of your customers or prospectives are coming in and saying, I really want to do biofluids, then you might be doing a better service instead of turning them away with maybe disappointing results from your mass spectrometer, saying, I can't do that throughput. Can you find a way to work with them to show them other technologies that could work really well for the scale that they're considering? And then now that they've had that like first taste of how cool proteomics is, now they're coming back to talk about proteoforms or to dive even deeper and find proteins that weren't in that panel with a smaller sample size that you could accommodate with your core facility. So these were some links. Again, we'll figure out how to do this. I don't know. But some links, if you just Google these terms, you'll probably find them as well. The business model canvas, the value prop canvas, and kind of the business assumptions exercise. It's called business, but every core is at some degree a business, right? We gotta pay for our doors to stay open. So with that, I wanted to save the last like 15 minutes for conversation.
Great, yeah, so we got 15 minutes to talk. I think we'll try to make slides available, maybe on the SPRG website.
Oh yeah.
And get you guys have access to these to show them to the people that are your clients, as well as to your administrators that are trying to say, okay, well, should this technology still exist in our university or whatever? So let's talk about these things. I think, Lindsay, do you want to keep up some of your slides?
Oh, I didn't, but I can pull them back up.
I have a comment to something you said, Lindsay, which was, well, these technologies can zoom in on a protein or set of proteins, and then the question becomes, of course, what are they doing, right? So I view this as a doorway into further analysis.
Yep, absolutely agree.
And it's that molecular level detail which does cause life to exist, and if we don't have that, then you're not going to understand it. So I just think that it's, if anything, it's your last slide about growing the market, and I think it'll be good, actually.
Yeah, totally agree.
So I have two related questions. First off, in a pure mass spectrometry lab, you can't do any of the readouts. And second, what about the cost?
So the readouts of alternative technologies?
I guess
Is that the question?
maybe the point is that maybe we're not supposed to do those readouts. Maybe the point is we're supposed to say, okay, well, there's another facility over here that can answer those questions. And then for that facility to say, okay, well, if it's a harder question than what they can answer, that they send them to our facility, our mass spectrometry facility. And I think that cooperation actually will benefit both candidates as the proteomics market increases.
Yeah,
You wanna-
and I think that's what John was getting at too, right? Building these strategic partnerships within the community so that if someone goes to SomaLogic because they want to do 3,000 samples, like our mass spectrometry, like that would take months for us to do 3,000 samples. They probably want something faster.
So Brian, from the informatics standpoint, who's going to own that? We as the mass spectrometry community have been, put our stake in the ground on how we pipeline some of these data. And each one of these other technologies has its own way to pipeline data. But which part of our community, how are we going to actually, when a biologist comes with a question, how do we answer that question? How do we feed the AI?
So I guess, let me unpack that. There's like several questions in one, right? So, classic.
Less of a question, more of a thesis.
I was going to say, at least another talk.
So from an informatics perspective, I think that there is a need for proteome informaticists to become more general purpose people that are focused less on proteomics technologies and more on general purpose technologies. But the informatics for those alternative technologies needs to get better for that to become a turnkey system, right? There have to be more people devoted to analyzing that type of data. That requires, I think, two things. One is more people, and the other is for vendors and companies to be more willing to open up their data for vetting, right? Those things are what are critical for us to propel this forward as a collaborative environment, right? We can, as mass spectrometrists, show how good our methods are in our circumstance. And I cannot take a SOMA data set and tell you how good this data set is and which of these measurements are reliable and which ones are not reliable. But if SOMA were to open up some of the underlying measurements, let me vet some of the individual reagents on their own, then I might be able to say in those contexts what's going on, right? So there has to be this give and take on both sides.
I think that also gets at the comment of the government should be stepping in to maybe help standardize this or at least better understand the structure of the data so that we can do more integrating it. We've already, I don't have super high hopes for integrating some of this data because we still haven't figured out how to really integrate multi-omics data in general, let alone within proteomics and different technologies. So my expectations are very low, but.
Yeah, thanks for coming back to that. I agree with Lindsay. I don't think that we're going to be integrating these different types of technologies in the same experiment, but they can answer different parts of an experiment. So sometimes you might use cryo-EM to study a structure of a protein, and you might use cross-linking mass spec to study a different aspect of how things get connected. I think we'll have circumstances like that where different projects, different parts of a project will be tackled by different parts. But the data itself don't need to actually be integrated in a clean way so that it presents the same measurement in both cases. Because I don't think that will ever happen.
What I don't really understand is that, all right, you're either going to send the sample off to the company or the project, but if you want to have it in-house, who can really do that? Because the readout crew is used to doing the genomics, so they do the capturing.
So if you have sequencing expertise, then potentially you could do it in-house. But what I'm arguing is you shouldn't try to do it in-house, right? You should try to let someone else do it because, as Lindsay says, there's so many different. The market is going to be huge, right? It's already, you see the number of proteomics companies that have been coming out over the last several years, right? Our segment of biotech is going to become much larger. And so there's not a reason why we have to be in charge of everything.
But I meant, let's say in-house in the university, is there supposed to be a core that can do that, or do you send it to the company?
So there are definitely, for certain of these technologies, cores that can do them. Not for all of them, right? SOMA, I don't think a core can run the technology.
You can.
Oh, you do? Okay,
set up
perfect.
a
Yeah,
site,
so there's room for other people to run other cores that are SOMA cores or Olink cores or whatever at the same university.
You have enough volume.
If you have enough volume. And what I'm saying is there will be enough volume if we allow these people in.
So I guess as a core administrator, I'd like to, maybe I'm wrong about this, but like to disagree with that, right? I think, wouldn't it make sense for someone to oversee proteomics and then there might be some stuff that's the non-mass spec portion of the team. There might be some stuff as a liaison outsourced to wherever. There might be some stuff put on mass spec, but with kind of an overarching view, you don't have you have less competing budget priorities and conflicts. And
Sure.
why should the mass spec team restrict itself to mass spec? Why shouldn't my proteomics director find someone who helps sort out?
I agree with that standpoint, right? Where mass spectrometry is part of this potentially much, much larger proteomics core, where we have other offerings. They might be in-house or they might be outsourced, and you have someone, one of these proteomics directors that helps connect people with your strategic
Someone who
partners.
understands the pros and cons and
Yes.
matching them up against the correct services, regardless of whether it's mass spec team, non-mass spec team, or teams, or as a liaison to external CRO.
Yeah, so I guess you can see this from multiple perspectives, and I think it depends on the flexibility of the core or the core director. And if the core director feels like they can sit up higher on bridging essentially multiple technologies, understanding what's going on with the mass spectrometer as well as with the sequencer, right? Then I think that actually works well. I would argue that it may be hard to find the people that can do that, and that's why thinking about it as there can be multiple cores that are collaborating together. That's another solution for this. Again, it depends on sort of the situation in your facility. If you feel like you can put them on equal grounds and not make it a mass spec core that does Olink on the side or Soma on the side, I think that's the important thing, right? These are equal partners that can work equally well together to tackle different aspects of the problem. Mark?
So, Brian, it seems to me that the strength of mass spectrometry is in discovery. We identify those molecules for which you want to make those antibodies or measure for some purpose attached to the biology that we understand. Its ability to do high throughput, many, many thousands of samples in the clinical lab is limited, clearly. So it seems to me trying to make mass spec participate across the entire board is not the goal here. We should focus on how we discover the science, the molecules that are important biologically, and feed those to the SomaLogic folks, to everyone else. I mean, in the case of SomaLogic, do you even know what you're binding many times? I mean, you've got the proteins that you put on the plate, and you've got your binders over here, but how do you know what proteins you're going to code onto your plate without that knowledge of the basic science? So why imagine that core laboratories should make processes that go all the way from the single molecule, single experiment detection to the clinical laboratory?
Yeah, I think that there's room for all of us on this stage, right? For everyone to have a say in this. And I would say that the things that as mass spectrometrists we should be letting go of are the cases where we have to deal with huge throughput, right? Where we're talking about multiple thousands of samples. Those are circumstances where you've got very clean proteins that you're interested in analyzing. Those are things that there are better technologies to try to deal with that specific problem. But I would argue that mass spectrometry works really well on both ends of that spectrum that are on the low throughput side, right? It works really well on quantitation with specificity for quantitation, and it works really well on detection for, again, specificity for what we're detecting. It's that sort of easy stuff in the middle that's really high throughput, that's where we struggle at. So I think that it's not like you should only use mass spec for discovery, and you should only use Soma or Olink for quantitation. I would argue that depending on what you actually need to do, what really the biggest issue here is the difference of scale and size, and what you're willing to kind of ignore versus what you want to actually interpret, right?
Agree.
I'm also happy to answer those other questions too, if we have time to chat later.
I'm just curious if there is any effort to benchmark the proteomics data with respect to...
Come up to the mic.
I was just curious that if there is any benchmarking effort is going on to understand the proteogenomics with respect to mass spec, and some of the language that each of these platforms are speaking to normalize them and say like, okay, we are talking same language, not like in India we have 100,000, 200 languages. It seems like proteogenomics and the proteomics field is almost on the same ground. Everybody is speaking every different thing. So, and the second thing is like how effective you think like this platform, especially proteogenomics, which is a sequencing-based platform, will be for post-translational modifications, like identification of PTMs when compared to the mass specs. I know that none of these platforms are providing yet that opportunity, but in future when that comes with those kind of things, how you see that is going to perform in comparison to mass spec?
I can answer the second one. You want to take the first one?
What was the first one? The benchmarking?
I'm terrible. The benchmarking.
Okay. There have been a couple papers. I know there's a SomaLogic Olink comparison paper, and there's been a couple papers comparing mass spec, but there has not, to my knowledge, been a universal benchmarking comparison of all of these technologies against each other. And I think I also wonder what the value of that would be. They kind of all specialize in their own areas, their own niches. So it's a little bit comparing like a screwdriver and a saw for hammering in a nail. Like, that's not the right test you should do. Like, you can benchmark that, but is it really useful? You would want to go get a hammer and hammer in that nail. And if you had a screw, you would want the screwdriver. And if you wanted to cut the board in half, you would go get the saw. So it's just choosing the right tool. Like, if I had a highly purified single protein, would I really want to run that on a mass spectrometer? It's a single protein. Maybe I would want to choose a different. A lot of these other technologies are built specifically for biofluids, and the proteins in their panels are not.
Looking at them, are you seeing the same sensitivity, each of those slices which you are analyzing with the mass spec? And what is the difference? Like when you should choose one method versus the other? That's the key question.
Let me dial that back. And I think what Lindsay is saying is that I don't think that the question works because the technologies are so different from each other that if you want to take Olink and or Soma and say, answer a question that can only be answered with mass spectrometry and try to pit them against our question, like PTMs, like you just mentioned, right? That's a hard problem for Olink or for Soma. If you try to pit them against that question, the answer is like, obviously mass spec is going to be better.
And I think they would admit that themselves.
Right?
Yeah.
It's going to be the same as. So, but in reverse. And so I think that there's no reason, in a sense, to say, okay, well, all these technologies need to be equally good at the same thing. It's really that each technology is good at the specific thing that it's good at, and we should specialize in using that assay in that particular way, in that type of experiment. All right, we got.
I think we're technically over time.
Yeah.
So I push back against that for a little bit. So I'm a clinical chemist. I run a clinical lab 90% of my time, and I have a core lab, which is mostly non-mass spec based, kind of these affinity stuff. So I'm very happy to sit in on this while my operations person is here to hear how awesome all this stuff is and why they should invest in my lab. But
you're welcome.
When
I'll
we
take my
do,
fee after our
yeah, when we see the our applications for studies for Olink, for SomaLogic, we're clinical hospital, research hospital, and we're getting hundreds to thousands of samples, and they're running it through this. And so this is a clinical correlation between disease status A and the results off of Olink and or SomaLogic and or Simoa and whatever else you want to throw it there. And because they're measuring different things, different molecules, I think those benchmarking are key, because there's just a lot of detail that's invisible to us, and we're using one word to refer to potentially tens or hundreds of molecules, and then we're doing correlations, and science junk in, junk out, right? So if we could figure out what exactly we're measuring in each one of these things, that would be very beneficial to, we would spend our money much more efficiently.
Okay, let me unpack that a little bit. So as a clinical facility, you're interested in measuring 1 protein, 2 proteins, 3 proteins?
Yeah, so
None of these technologies are the right way to think of this, right?
So I have a core lab,
Okay.
right, and then I have a clinical thing, and I don't think we're going to. I don't expect that in 10 years I will have single biomarker assays. I think I'll have Olink. However big a plex I can do, and be absolutely quantitative, or at least be good at clinical specificity. That's what I'll be running.
Okay, so again, as we've been talking about, we don't know what these molecules are binding to in many of these affinity assays, right? And so to use one in a clinical setting, I think would be terrifying, right? If we don't actually know what it's doing. And so from that perspective, I think that the vetting needs to happen at a very different level than what we're talking about here with just saying, can we get reproducibly the same number out, right? You can get reproducible binding to background, right, to a totally different protein. That doesn't mean that you're actually measuring the thing that you're interested in measuring, or that what you're looking at is an off-target effect, right? So I've been working with another research group that have been using IHC as their main readout for protein molecules to say that there was something going on with this particular set of samples. Because they're using an antibody as their base, this was sort of the center of the field for 20 years, was using this one set of antibody suite to say that this thing was going on here. Turns out that none of it was actually going on when we analyzed with the mass spec, right? Because we were seeing off-target effects for all of these antibodies. They just happen to correlate well with each other, right? We can't trust that we have these affinity binding work in those circumstances unless we know what it's actually binding to. For that to happen, the companies need to open up their reagents.
I want to add some of that, Brian. So the amount of work that goes into a single biomarker ELISA assay, including dilutions and matrices and all that sort of stuff, for an actual clinically applied ELISA is vastly in excess of anything I've seen for any of the, I'll just call them 3D binding assays.
Yes.
Right? And the point is that you have this menu that can have all kinds of stuff in it, right? Can you get an interfering molecule? Yeah, and an endogenous autoantibody is going to prevent detection by any of those, and for that matter, by an ELISA as well, which is why LabCorp went to a mass spec-based assay where they ingested everything. So there's a huge amount of work to be done on that. What you just mentioned, what you guys both mentioned, made me think what we need is a SomaLogic array of columns. I'm serious. Let it go through, right? Wash it out. Do mass spec. 7,000 samples is annoying, but that would get at the answer.
So we're 5 minutes over. This has been a great discussion. Thanks for staying this long, and I look forward to talking to the rest of you at the conference. All right, thanks.
Great job.
I'm Lorianne from SomaLogic. I want to give